can hear me. So welcome, and it's a pleasure speaking to you all today about our new innovative technology to build tissue models. So I will take you through a series of slides. Will be about 30 minutes or so, and there'll be questions at the end if you have any, as Bob has said. So by way of overview, I will give you some background as to why, first of all, 3D cell culture is important, and then how we use 3D cell culture as a technique to engineer tissue equivalents in vitro. So I'll give you an overview of our technology, the products that we have available to you to enable you to do this. And then I shall go into a deeper dive into a couple of example applications of where we've used this technology to build models of human tissues. Okay, so without further ado, we'll commence the presentation. So the microenvironment in which cells grow in the lab can substantially influence cellular behavior. And the point of this slide is to show you how a cell, as depicted in the center, can be responsive to all those different factors, which we can also control in that microenvironment. So things like the nutritional status of the media, what you add to the media, the fact that you can introduce morphogens and hormones, the cell matrix adhesions and the extracellular matrix components, so coatings of plastic, for example, the connections between cells and their signals, mechanical stress, and importantly, the three-dimensional physical space. And all of these factors can influence how cells behave, and of course this can take place over time, as depicted by the clock. So I'm going to focus in particular, the 3D physical space. So if we consider how a tissue looks, so in vivo along the top line, we can see very clearly cells residing in a complex three-dimensional environment, and that's also depicted in the schematic, and as a single cell. And you can see that single cell in relationship to its neighbors and also the extracellular matrix. And that's what we would find in a tissue, and of course there'll be multiple cell types present. Now in vitro, particularly in conventional 2D culture, we break down those relationships between cells and we introduce them into this foreign microenvironment, and they encounter a flat planar substrate, and those cells will change their shape and form. And as they do so, they will remodel their internal cytoskeleton, which can impact on major organelles such as the nucleus, which in turn can affect gene and protein expression and therefore function. So a cell which is adapted to a flattened two-dimensional configuration may well have different behaviors to their native counterparts, and this in turn can influence the behavior of that cell in response to drugs, or what questions you're asking of that cell culture. So it's really important to be aware of this. You can see this very clearly if you look at a single cell. This is on the left, we have a fibroblast, which has been stained with phalloidin, which is an F-actin stain. And you can see how thin that cell has become in 2D, whereas in 3D, it's three-dimensional all the way around. And you can also see on the right how the major organelle, the nucleus, is also influenced and changes its shape. And it is of no surprise that this change of shape can influence, as it says in the papers published at the bottom, gene and protein expression. So this is a long established phenomenon. People have known about this for many years, but we still grow our cells in 2D culture, and it's important to be aware of this limitation. And this has given rise to, partly, the reason why we now have enabling technologies that allows cells to grow in 3D to overcome that problem. And we can use those technologies as a technique to also bioengineer human tissues. And I will come on to that in more detail as we go forward. So the point of 3D cell culture is to recreate a more natural environment for three-dimensional growth, to enable us to get closer to a real tissue situation. And as a consequence, we can enhance structure and function of the cells and create more representative models, which hopefully will produce more improved data with greater predictive accuracy. This is important because it has extensive impacts in certainly the discovery process for research and also product development. It can also influence the efficiency of the discovery process and saving time and money. And of course, humanized three-dimensional tissue models can also replace and reduce the numbers of animals used in research. So we set about developing a technology that can enable us to grow cells in 3D and build tissue in vitro. And it had to be a technology that enabled us to be able to do this routinely and robustly. So this is a scaffold-based approach. And when we started to develop this initially, we decided that it had to satisfy various attributes, such as those listed here. So for example, the material that we made our scaffold from is inert, and it does not degrade and remain stable. And this is important because if it was a degradable material, it could fall apart and become physically unstable, but also it could release byproducts into the culture which themselves could influence the behavior of the cells. So we didn't want that as an additional variable. In fact, the material of our scaffold is the same material as an existing two-dimensional cell culture product, which of course are made of polystyrene plates and inserts, etc. Our scaffold is made of polystyrene, and it is adaptable to existing cell culture formats. So these are industrial standards such as multi-well plates, well inserts, and they are all developed as consumable products. So let me now introduce you to the scaffold membrane itself. So across that top line, we see 2D culture. So these are conventional plates, cells growing in 2 dimensions as monolayers. And what we've done is we've changed the geometry of the polystyrene to create this highly porous material. It's actually 90% porous, meaning that 90% of that material is actually space in which the cells then occupy. We can see cells entering this material and growing in 3 dimensions within the XYZ configuration. We've taken that material and we've engineered it into a 200 micron thick membrane, and you can see that here by the scanning electron micrograph, higher magnification in the central image here. The image on the right at the bottom is actually full of cells growing in 3D within the membrane. That's why you can't see the actual scaffold structure. And in this case, what we've done is we've created a 3-dimensional slab of cellular material. As I say, it's 200 microns thick, so the cells centrally are only 100 microns from the source of nutrition. So that material has been commercialized into a product called Alvetex. Alvetex scaffold is available for purchase commercially via REPROCELL and other distributors. And what we see on this slide are the various formats for these products. So along the top line, we have multi-well plates. And you can see we have different configurations, 12, 24, 96, and for well. In addition, we have well inserts, 6, 12, and 24. We also are in the process of developing 96 well prototypes, as depicted in this image, for higher throughput applications. So these are the products which are available. As I say, if you go to the REPROCELL website, you will clearly see the different formats on the online store. So let's just have a quick look at how cells behave in the material. So the question is, does the material prevent that shape change that we see in 2D culture, as I've described? So this top line here shows how a cell flattens. We can see that very clearly in side view in the 2D situation, and the nucleus as well flattens, whereas in the Alvetex material, the same cell type does not change shape and retains a three-dimensional structure. The presentation of the scaffold material is also important. And that's why we offer different product formats. So if we consider scenario A, we see here the scaffold full of cells being located in the bottom of a well, of a multi-well plate. So in this situation, the cells receive nutrition from the medium above only. And you could argue that actually the cells at the bottom are at a different level, and that they do not get the same nutritional requirement, and they could build up lactic acid, for example, and have less access to oxygen. Now that could be an advantage for your particular interest if you perhaps want to model certain situations that you would find in, say, a tumor. If you don't, and you want your cells to be more healthy and viable throughout the material, then you might want to consider the well inserts. So the well inserts are our top selling product, particularly the 12 well insert. And this is depicted in this scenario in B. And we can see here the membrane suspended with compartments of media above and below. And the cells can receive nutrition, as I say, from both media compartments. The other thing to consider is that when you're growing such cultures in 3D, you have many more cells per unit volume of medium than you would normally in a conventional multi-well plate. So to get around this issue and to maintain a high quality of your culture medium throughout your experiment, you can either change your medium more regularly, or you can introduce a greater volume of medium to your 3D culture, as depicted in scenario C. And we have what we call a deep well Petri dish, and this shows an example, or this photograph shows an example of this product, housing 3 6 well inserts in this case. So a large volume of media to support these 3D cultures over time. There's also the opportunity of designing experiments for the culture of multiple cell types. And because of the nature of Alvetex and the different product formats, there are various ways in which this can be done to help customers be able to design their experiments for their particular needs. So for example, imagine the scaffold in the bottom of a multi-well plate, and here we have 2 different cell populations. You could also have the situation in a well insert, or you could have, as shown in assembly option 2, a well insert in 3D with a 2D monolayer of cells, and you could study paracrine signaling between those 2 populations. You could do the same thing in 3D or in 3D in contact directly, which could also be beneficial for cell communication. And it's assembly option 5, we've got 3D culture, and then we've seeded on top of that an additional cell population. And these are just some examples of what can be achieved using this technology. So let's then look at how we've gone through achieving some of those scenarios. And what I'm going to do now is focus on how we've used this technology to construct mucosal tissues that you find in the body. So a mucosal tissue, by definition, is an epithelium that sits on a basement membrane with underlying supporting stromal cells. So in the schematic here, we have that depicted. We see the epithelia on the surface, the basement membrane, and then underneath we have the submucosal compartment, which consists primarily of fibroblasts, but of course, in reality, you have other populations of cells present, for example, blood capillaries, lymphocytes, nerves, etc. So what we're interested in doing is using this Alvetex technology to be able to recreate those structures in vitro in a robust and reproducible manner. And I've got 3 examples to share with you today. We have a simple columnar epithelium, which would typically be found in the intestine. We have a stratified non-keratinized epithelium that's found in the mouth, and a stratified keratinized epithelium, which is the skin. And of course, these all have different functions and different structures. And that's really important to remember, because structure is related to function. And what we spend a lot of time doing is ensuring that the structures that we produce in vitro align as closely as possible to what we see in the real tissue in vivo. So we compare side by side continually. I shall show you some evidence of this. So first of all, let's look at the skin. So this is human skin, as shown by this histological image. We have an epidermis sitting on top of the dermis. Now the epidermis is complex in that it consists of keratinocytes at different stages of differentiation as they move towards the surface and produce a stratum corneum, where of course they are then shed. In the dermis, we have primarily fibroblasts, as I've mentioned, and we also have a lot of extracellular matrix, primarily types of collagen, but also elastin, fibronectin, etc., depending on the tissue type. So first of all, to produce skin, what we do is we use primary fibroblasts. We take those fibroblasts from explants of patient material, and we explant them into culture, and then when we've generated sufficient cell populations, we will then seed them into the Alvetex membrane. And we're doing this in a well insert. This can be a 12 or 24 well or 96 well insert. We give those cells sufficient time to enter the material, populate it, grow on the surface, and also deposit significant quantities of extracellular matrix. And we can monitor that by various methods. So for example, on the left, we have immunofluorescence detection of various extracellular matrix markers, such as different isoforms of collagen and fibronectin. We can also perform biochemical assays on these materials, as shown on the right, where we're measuring total collagen deposition over time inside the Alvetex membrane using a hydroxyproline assay. So the deposition of the ECM within the matrix is important, and that has to happen over, we grow these cultures for 4 weeks to establish what we call a foundation. So that foundation is comprised of the fibroblasts and the ECM within and on the surface of Alvetex. Once that's been established, we can then seed onto the surface the keratinocytes, and we then raise them up to the air liquid interface, where they can then differentiate, forming a stratified epithelium and a stratum corneum. And this can be maintained for many months in vitro. And we show here, for example, up to 28 days. So this is where we produce our fully formed full thickness skin equivalent. And I can show you this on this slide, looking at the histology. So we have very clearly the dermal compartment housing the Alvetex membrane, and on the surface at the air liquid interface, we've produced a clearly stratified and keratinized epidermis. You can see very clearly the different layers within this material. We can do this reproducibly using different batches of cells from different patients. We can produce quite large pieces of skin in this case, as you can see, several millimeters in diameter. And we perform in-depth characterization. So as I mentioned before, we compare our in vitro model with human skin material side by side. And what we're doing here by immunodetection is looking at various protein markers in the in vitro equivalent compared to real tissue for epidermal differentiation markers, epidermal intracellular junctions, and also dermal extracellular matrix proteins. And this level of characterization demonstrates that the bioengineered tissue model on Alvetex compares very favorably to real tissue. We also have looked very closely at the ultrastructure of these models using transmission electron microscopy. So on the left, we can see a fibroblast, which is within the Alvetex membrane, and it's producing lots of extracellular matrix. So this is all human-derived extracellular matrix from these fibroblasts, and this is why it takes time for this to happen. And it's really important that that does happen, because if you then seed your cells too early onto these foundations, then you will get invasion of the epithelial cells into the matrix. So we want to avoid that, and we found that 21 to 28 days of growth of the dermis is sufficient prior to the growth of the keratinocytes on the top. On the left, on the right, what we can also see is the basement membrane, as shown by these asterisks. And that's really important because the basement membrane and the hemidesmosomes are located here, and this is the interface between the dermis and the epidermis. So more data. This is now scanning electron microscopy. So you can see on the left the transverse section through the Alvetex. These are where the fibroblasts are located, the epidermis on the surface, and then looking directly from above, you can see the stratum corneum. These are the corneocytes, the flaking cells lost off the surface of your skin. So it's directly recapitulated. And then on this slide, we've got some more TEM analysis looking at this. In this instance, the desmosomes. These are electron-dense structures, and these bind the cells together and help with the physical integrity of the model. At the bottom, what we have is a functional assay to challenge the barrier properties of the skin model, where we are using fluorescent dyes, an exclusion assay in the presence of increasing concentrations of a detergent, SDS. So as the SDS levels start to increase, eventually it overcomes the integrity of the barrier. So that's a very useful assay to demonstrate how strong the model is. And actually at 0.25 and 5.5% SDS, that's quite a strong detergent. We're now looking to increase the complexity of these models, and what we're demonstrating here is the addition of multiple additional cell types. So we've already got the keratinocytes present in this epidermal model, and now we're introducing melanocytes, so we can get skin of different tones. And in the bottom here, we've got an example of introducing immune cells into the dermal compartment. This is ongoing work to enable us to create an inflammatory skin model. So let's look a little more closely at the melanocytes in the full thickness system. So we've got here melanocytes stained with an S100 marker. The cells reside very nicely at the basal location in the epidermis in the full thickness model, as they should do. You can see very clearly the pigmentation of the skin. And if you look even more closely, you can see in real human skin the formation of what we call the supranuclear caps. So this is where the melanosomes crowd above the nuclei of the keratinocytes, and it's hypothesized that this is to protect the cells from UV radiation, for example. And we can see exactly the same thing has been recapitulated in our in vitro model. So we see here very nicely in melanocyte, the melanosomes within it, and we can see a very nice example of a supranuclear cap, and that's all very clearly shown by the transmission electron microscopy of the in vitro model. So all of this is achievable using this Alvetex platform. Here's another example where you can develop, in this case, a disease model. So we have our skin system, full thickness model, where we've introduced melanoma cells, and these melanoma cells, the interface of the epidermis and dermis, can then invade into the dermal compartment. And this is a model of cell invasion in 3D within this tissue model, and we can compare what happens in vitro, so these two columns here on the left compared to the real situation in pathology. This is melanoma in situ and metastatic melanoma, and it enables you therefore to model certain aspects of the invasion process in a tissue model in vitro compared to the real situation. And then finally for skin, we've got a situation where we can recapitulate the aging process. So using cells from aged donors, for example, we can create models where the skin epidermis is a lot thinner and there's less extracellular matrix, particularly collagen, in the dermal compartment. And this is exactly what happens during aging. So these are very useful models for different applications. So that's just a summary slide. If you're interested in reading more about this particular model, there is a paper, and that's published in the Journal of Anatomy. That's the front cover of that journal, where we speak about the bioengineering of skin. So let's look at a different example, and this is bioengineering the human intestinal mucosa using the Alvetex platform. Now this is different because here we're not using entirely primary cells. We're actually using also a cell line, and I included this to demonstrate to you how cell lines, which are commonly used in industry, can be altered by growing them in 3D, particularly in co-culture with their supporting fibroblasts. So let's look at the structure of the gut wall. So here we have a simple columnar epithelium sitting on what is called the lamina propria in the intestine. And that's composed primarily of fibroblasts. But of course, as I say, there are other cell types present. So the current technology to assess intestinal function in vitro primarily involves the use of this cell line called CACO2, which is traditionally grown on millicell transwell inserts in a 2D culture. And you can see that example there. This is an overly simplistic situation. The functional characteristics of this epithelium are recognized as being abnormal, and it's a model, and we can do better. And I want to demonstrate to you how we can do better using our technology. So what we do is we use fibroblasts derived from colon, CCD18 cells. We put them into Alvetex just as we did before. We allow them to grow, and then we seed onto the surface CACO2 cells. What this slide shows at the top is, first of all, the formation of that submucosal compartment, again with the deposition of extracellular matrix over time. And then when we seed our CACO2 cells onto the surface, they change their shape and they stand up, they become columnar, they are polarized, they have a very prominent microvilli at the top and a basement membrane, and tight junctional complexes throughout. And I shall show you some more data in a moment on those complexes. So what we see in this slide is histology of this model over several weeks, and note the consistency and reproducibility and how it can be maintained for long periods. So this is electron micrographs. So we've got scanning images of the submucosal compartments at different magnifications. This is essentially the foundation upon which we then see the CACO2s. CACO2s form this cobblestone morphology. You can see very clearly the microvilli and the glycocalyx on the surface. And these transmission electron micrographs again show microvilli, junctional complexes, and the basement membrane at the bottom, and the basal aspect of the epithelium. We compare the in vitro model with the in vivo tissue, just as I said, for a variety of different markers. So within the matrix, to check the deposition of the ECM. We're also looking at various junctional complexes such as occludin in this example. So we can create membranes of this material as depicted in these slides here, several millimeters across. And we take those and we put them into ushing chambers. So this ushing chamber in the central region here is where the membrane would be located. And normally one would put in animal tissue there, for example, from rat or rabbit. And then you'd have a chamber either side, and you could study the passage of drugs or ions across that membrane. So we can, of course, now do that with our 3D humanized tissue model. We can also measure short-circuit currents using this electrophysiological apparatus as shown in the photographs on the left. So we can generate lots of interesting data. So interestingly, tear values in CACO2 are known to be notoriously high, but in our model we see the tear values come down significantly, and we also see changes in junctional complex expression. We also can then use these models, as I said, for testing of drugs in terms of initiating ion fluxes, absorptive responses, etc., and studying transporters within this model. We're also, in this model, building greater complexity as we did with skin. So we're now introducing goblet cells into the system, as you can see them here, and the goblet cells into the model. But we're also now introducing into the submucosal compartment immune cells as a step towards developing a system to study inflammatory disorders of the gut, notably IBD, Crohn's, and ulcerative colitis. So this is again an example how Alvetex technology has been used to create sophisticated tissue models. And the last quick example I'm going to show you is the oral mucosa for oral healthcare applications. So again, we are able to follow the same strategy. So in this case, we're putting fibroblasts from the mouth into Alvetex and allowing them to grow to form collagen and other extracellular matrix within that construct. Again, creating this foundation to support the surface epithelium, which we then seed onto the surface. And this time it stratifies, but these primary epithelial cells, they do not keratinize, because this is the inside of the mouth. So it's a non-keratinized stratified epithelium. Again, we compare that to real tissue as shown on the right. So as I say, this is an example of an application for oral healthcare. So to summarize, Alvetex Scaffold enables you to perform three-dimensional cell culture experiments, but it goes beyond that as a technique. It enables you to bioengineer human tissue models of the types that I have shown to you today. But we've only done these particular ones. Other customers have produced other models for their own needs, having bought the product and used it for their particular requirements. And if you go onto our website, you'll see some of the publications which have been produced by other customers. For example, a very nice model of the human respiratory lining. Okay, so in summary, this technology enables us to recreate a more natural 3D environment, bringing us closer to real tissue. The environment in which the cells grow play an important role in their structure and function. Alvetex Scaffold enables us to create these more sophisticated culture systems, and this is a rapidly advancing field which will have significant implications which will benefit research, increase the efficiency of the discovery process, and reduce animal usage. So if you'd like more information about Alvetex and the protocols and various application notes, white papers, and publications, please do visit our website at REPROCELL.com.